Good day, grade 12 learners. Welcome to today's life sciences lesson. My name is Hotato Litlotlo. I'm not alone today. I'm with my colleague Suki. This lesson is brought to you by the Department of Education in collaboration with Saibono Discovery Center. Remember, now we are busy preparing for the big day coming tomorrow. So we need to make sure that we are prepared for the 75% that you need to make sure that you get most of it. I know that some of you, life sciences may be a challenge to you, but what I want to tell you today is that preparation, and preparation, you will never go wrong. For you to pass life sciences, you have those learners that are struggling with life sciences. Section A must be your section. Just make sure that you do very well in section A. Also for you, a distinction candidate, for you to get the 80, the 90, and even 100% in life sciences, make sure that you excel in section A. Remember, be on time, relax, and do not panic. So today's lesson is paper 1 revision. So let's have a look at what is it that you need to know. By now, you should have started studying. Let's have a look at what is it that you need to know for you to be fully prepared for tomorrow. Remember, today's lesson is not necessarily about reteaching content. We are just going through the content that you need to make sure that you know before tomorrow. So we have resources. You can find us in Instagram. You can also find the resource link in our Google Drive. This is not the time to be looking at so many resources that can actually confuse you. We have voice notes that nicely summarizes concepts. We have short video clips that we have sent to your teachers to forward to you. Those video clips were made by our provincial coordinator, Dr. Griselda. So make sure that you actually get hold of those video clips. You look at all the difficult concepts that were challenged during the prelims or during the course of the year. Those concepts are covered using those video clips. Terminology is very key. Terminology is very crucial. Before you start studying, go through your terminology because it is going to beef up your content level. So make sure that you use the updated terminology list. Where you're going to find all these resources, grade 12s, in your learner resource folder, the Google Drive. Having said so, this is the content for paper 1. If you look at the topic responding to the environment, is 54 marks out of 150. So that simply means when you start studying, when you start preparing grade 12, make sure that you start with the topics that have most marks. Looking at responding to environment in humans and also human reproduction. Those two topics, they are close to 100 marks out of 150. Then also another important topic, all the topics are important. Another topic that has most marks is endocrine and homeostasis. And one thing beautiful about the endocrine and homeostasis, already you have a background from grade 11. Not forgetting reproduction in invertebrates, as much as it only has 8 marks, but it is also important. Human response to the environment. What is it that you need to learn and make sure that you excel before you actually go to the examination room tomorrow? Let's have a look at it. We have what we call the examination guideline. We are still using learners, we are still using the examination guideline of 2021. The examination guideline shows you the content that is examinable at the end of the year. Our textbook will be loaded with so much content, but the examination guideline shows you that this is the concept that you need to learn and also the depth in which you need to learn that concept. So let's have a look at the responding to environment humans. What is important here is that these are the structure or the parts of the brain. We have too many parts of the brain, but you need to know the structure and function. Having said so, when it comes to life sciences, it is about structure, be able to label, know the functions, beyond the functions, know the structural suitability to say, how is this structure adapted to perform its function? We'll get to that as well. We normally call that the cause and the effect. Beyond that as well, grade 12 learners, you need to know the structural dysfunction, or what happens, or actually the structural failure. What happens if a certain structure cannot perform its function? What will be the consequences? So the part of the brain that you need to be able to know by tomorrow: cerebrum, the function and the structure, where are they located? Cerebellum, and normally learners, you confuse cerebrum and cerebellum. Medulla oblongata, hypothalamus, and corpus callosum. You need to know the spinal cord, the structure of the spinal cord, and also to be able to describe the reflex action using a diagram. You need to be able to draw the 3 types of neuron that we have: your sensory neuron, your interneuron, and also your motor neuron. Remember, we're no longer using the term connector neuron. The human eye, know the structure and functions of different parts of the eye. There are processes that are very important as well that pertain in the eye, that is accommodation. Be able to define what is accommodation. That is why in the beginning I said to you, terminology is key. Know your terminology. Section A and some of the short questions of section B, it's all about terminology, the pupillary mechanism. And when it comes to accommodation and pupillary mechanism, you need to be able to know which structures, which muscles are responsible for accommodations, which muscles are responsible for pupillary mechanism. And the last one is the human ear. The same thing applies. You need to know the structure of the ear and different functions of the parts of the ear. There are processes that are important as well, which is the process of hearing, balance, and hearing defects. The nervous system. Remember now, grade 12, the nervous system is divided into what? It's divided into the central nervous system. Whenever we speak of the central nervous system, we are speaking of the brain and the spinal cord. We have what we call the peripheral nervous system, and normally we're not you are not used to the how is actually shown here to say the sensory pathway and the motor pathway. Whenever we speak of the peripheral nervous system, we normally ask you what forms components of the peripheral nervous system. That will be your spinal nerve and your cranial nerve. Another important one is the autonomic nervous system. Autonomic nervous system is divided into what? Into the sympathetic and the parasympathetic. So remember those as well. Remember when we speak of the sympathetic, what increases the heart rate. When we speak of parasympathetic, what decreases the heart rate. Having said so, grade 12s, now let's have a look at the location and functions of different parts of the brain. As I've indicated, learners, it is very important of you to know the different structures. Don't just know the structure, know where the part is located in the brain and what is its function and how is it structurally suited to perform its function. We'll come to structural suitability when we deal with other parts of the concepts or the other parts of the syllabi. Then what is important here is that, as I've indicated, these are the types of brains that you need to know. Your cerebrum, what is the function of the cerebrum? And remember, they are not gonna, it's not all the time whereby they will ask you what is the function of the cerebrum. They will give you a scenario. The scenario can go as far as the learner had to memorize maybe cell phone numbers. You need to know that which part of the brain deals with memory, that will be the cerebrum. Which part of the brain deals with controlling the breathing rate, that will be the medulla oblongata. So please make sure that you know the different parts or the different functions of the brain. And as I've said, do not confuse cerebrum and cerebellum. Moving forward, let's have a look at the 3 types of neuron. It is very important, practice, and practice always makes perfect. Make sure that you are able to draw the structure of the 3 different types of neuron. That is your motor neuron, the sensory neuron, and the interneuron. And one of the easiest way of remembering this, especially the difference between the motor neuron and the sensory neuron, is one of the possible questions learners that they can ask you to say, What is the difference between the motor neuron and the sensory neuron? The easiest way of remembering, look at the position of the cell body. The position of the cell body is a giveaway. And always when you are requested to draw, your caption must be there. Especially when it comes to the neurons, you must show the direction of the impulse. That what? The impulse is transmitted away from the cell body. So it is very important to show the direction of the impulse, and also the important thing also is the labels. You need to know where are the dendrites located and what is the function of the dendrites and what is the function of the axon. Remember now the function of the dendrite, the transmission of impulses towards the cell body, and when it comes to the axon, the transmission of impulses will be moving away from the cell body. So it is very important, grade 12, to know how to draw this neurons, and also to be able to differentiate between the motor neuron, and also to be able to differentiate between the motor neuron and the sensory neuron. Let's have a look at terminology. The reflex action and the reflex arc. Learners, you normally confuse the two to say what is a reflex action. In life sciences, there are key words or key terms that you need to always, or they need to be always part of the definition. Whenever we speak of a reflex action, we are speaking of a rapid, automatic, someone can say involuntary response to a stimuli. And remember now, you need to understand what are we talking about when we're talking about the stimuli. That's when now we talk of any internal or external change in the environment. That is a stimuli. So a reflex action is a very quick automatic response to a stimuli. One thing that learners you tend to do also here when it comes to the definition of a reflex action, it is not autonomic, it's automatic. So please do not confuse the two terms. So that simply means we are referring to all those actions that are controlled by the spinal cord, that do not go via the brain. And also, what is the significance, what is the importance of the reflex actions? The importance of the reflex action is, it says that it allows for a quick response to a stimuli without thinking. Why is that important? So that the body can be protected from harm. You cannot just generalize, learners, and say it prevents damage. What damage? You need to be specific. That is how most of our learners, you are losing marks, because sometimes when you answer questions, you are answering questions not based on the context that you are given. You are just giving a general statement. So be mindful that when you answer questions, read the question thoroughly before you provide an answer, and do not be too excited as well or too nervous, because you tend to make mistakes. And remember here, we're talking about then what is a reflex arc? Remember, we said the reflex action, we're talking about a rapid, involuntary response to a stimuli. A reflex arc now, think about the path. In order to bring about a reflex action, the nerve impulse must be transmitted along a path in order to bring about a reflex action. So it is the path, an arc is the pathway that a nerve impulse is transmitted. Some learners will say it is a pathway that the message travels. It is incorrect. Life sciences, it's a science subject that has its own terminology. You cannot mistake an impulse with a message. So correct terminology is key. So with the reflex arc, we are talking about a pathway along which an impulse is transmitted to bring about a response to a stimuli, or one can say to bring about a reflex action. As I've indicated, do not confuse the reflex action and the reflex arc, and avoid at all costs, actually avoid by all means to use message instead of a nerve impulse. Now let's have a look at the diagram that is reflected or projected here. Always remember, learners, that whenever we speak of a receptor, irrespective where that receptor is found, whether we'll be talking about the photoreceptor, the cristae, the maculae, or we're talking about the cold or the heat receptors, the function, the main function of the receptor is to detect a stimuli and convert that stimuli into a nerve impulse. That is a basic function of what? Of a receptor. So what happens here is that the pod is hot. So if it happens that you accidentally place your finger on the pod, what is going to happen? The receptors are going to pick up the stimuli, which is what? Which is the heat here. And that stimuli is going to be converted into a nerve impulse. The nerve impulse will be transmitted to the central nervous system. By which neuron? By the sensory neuron. So always remember, grade 12, whenever we speak of the receptor and the sensory neuron, they are best friends. They are linked. Receptor, sensory neuron. Effector, motor neuron. And whenever we speak of the sensory neuron, think of the dorsal root of the spinal cord. Motor neuron, the ventral root of the spinal cord. And another important thing is that, as I've said before, read into what you are given. Analyze the question that you are provided with. The context will always change. Just because you see the structure of the reflex action or the structure of the spinal cord, then don't just assume that, oh yes, they're asking about the reflex action and you know that it will be from the receptor to the sensory neuron, spinal cord, then interneuron. Not all reflex actions involve the interneuron. So please read into the question that you'll be provided with. Then we know that the sensory neuron will make a synaptic contact with what? With the interneuron. And in turn, the interneuron will also make a synaptic contact with the motor neuron. The motor neuron will transmit the impulse to where? To the effector. That's when and in this case, the effector we're talking about, you need to be specific based on the question that you are given. In this case, the effector we're talking about what? The muscles. Then the muscles of the hand is going to contract and move away and pull away from the hot pot. So that is the reflex arc, or that is the reflex action. So please make sure that you study the concept and you understand the different parts of the spinal cord, and you need to be able to understand that what enters through the dorsal root is the sensory neuron that is transmitting impulses. That is, the impulse is picked up from where? By the receptor. Lovely, grade 12 learners. Remember, tomorrow, arrive on time. Arrive on time. The reading time is very crucial. That's when you get to plan how are you going to answer the paper. Now let's have a look at one of the previous year's questions. That was the May-June of this year. The diagram below represents the part of the reflex arc. Just take a minute, analyze the diagram. What is it that you're given? You are shown that there is a part there, a receptor, there is a neuron, and that neuron, you can tell that it is entering the spinal cord. You have neuron E as well. You have neuron D. You have another part at the bottom, the ventral root, and you have an effector. The first question says, give the letter of the part that represents A, the dorsal root. Which part represents the dorsal root? That will be B. And which part represents the sensory neuron? Remember, I said to you, always remember, receptor goes with what? With the sensory neuron. So, and the sensory neuron is the one that transmits what? The nerve impulse to it, to the central nervous system or to the spinal cord. So that simply means our answer will be B and an A. State the significance of a synapse. Terminology, very important. What is the importance of a synapse for 1 mark? And also look at the mark allocation. They will guide you in terms of how to structure your question. Because here, they are just saying to us, we must state the significance. But if they say to you, explain the significance, that is something else. So state the significance of a synapse. It ensures that impulses, not messages, but impulses. It ensures that the nerve impulse are transmitted in one direction. Let's move on. Explain the significance of a reflex action. This one, grade 12 learners, this question, it's a cause and an effect, because now they're saying you must explain. They're not saying what is the significance. Oh, they're not saying to us, state the significance of a reflex action. They're saying to us, explain the significance of the reflex action. So now you need to start with what? With defining this reflex action, the reflex action, to say it is a rapid automatic response to a stimuli. Because it is a rapid automatic response to a stimuli, it is able to protect the body against damage. We are not saying it is it serves as protection. We are specific that it protects the body against what? Against injury, or someone can say against damage. So it's a cause and an effect. Hope you'll remember that tomorrow. Explain on the reflex arc if neuron A is cut. Remember, I said to you what happens when we have a problem in a certain part of the structure. Neuron A, if we quickly go back, neuron A we said is the sensory neuron. So now, what will happen when the sensory neuron is cut? Grade 12 learners, think of the function of the sensory neuron. What is the function of the sensory neuron? The sensory neuron function is to transmit impulse from the receptor to where? To the central nervous system. So that simply means that function, it won't be able to perform that function. Then you need to tell us that impulses won't be able, the impulses won't be able to be transmitted from the receptor to the central nervous system. Then in turn, what is going to happen? No impulses will be transmitted from the receptor to the central nervous system, or to be specific here, to the spinal cord. Then that is a cause as well. It's a cause and an effect. Then since impulses won't be transmitted to the spinal cord, what will be the effect of that? The effector will not be stimulated. One can even take it from the motor neuron won't be able to receive the impulse. Then the impulse won't be sent to what? To the effector. That simply means the effector won't be what? Won't be stimulated. Draw a label diagram to show a detailed structure of neuron D. Neuron D, we're talking about what here, grade 12 learners? We're talking about a motor neuron. Remember, I said, always remember the position of the cell body. Right at the top, that's where you find the cell body of a motor neuron, unlike the sensory neuron, where the cell body is more or less in the middle. So this is how we mark this, the structure. The diagram of a motor neuron is one mark. Then we look at, did this letter draw a motor neuron? Yes, it is a motor neuron. The correct caption, you have indicated motor neuron. Any 3 correct labels. Do not limit yourself. Normally, the norm is we will ask for 3 labels, but do not limit yourself to only 3 labels, because you might get 1 label incorrect. Look at the position of the myelin sheath, cytoplasm, the dendrite, the cell body, the nucleus. From the previous question papers, we know that we have the terminal ends of the axon. They hardly give a mark for that label. Actually, I've never seen any previous question paper where they've given a mark for the terminal branch of the axon. Lovely, grade 12s. Moving on, now let's look at the internal structure of the eye. Beautiful concept as well. Beautiful concept as well. As I've said before, grade 12s, know the location, know the structure, know the function. You need to go beyond knowing the function. If you really need that A in life sciences, or a good mark in life sciences, you need to understand what happens when that structure is not able to perform its function. You need to ask yourself, how is that structure adapted to perform its function? That's when now we speak about structural suitability. From the optic nerve, know the function of the optic nerve, know the function of your yellow spot, your fovea centralis, the retina, your choroid. Do not confuse the choroid, the layer of the eye, or the layer of the eye that is richly filled with blood vessels, that is dark. Remember, the choroid is that layer of the eye that is richly supplied with blood vessels, and it supplies the eye with nutrients and with oxygen. It is also responsible for absorption of excess light. Do not confuse that layer choroid and the chorion, which is the outermost embryonic membrane, because the terminology or the terms are a little bit similar. So it's choroid and chorion. So do not confuse those two. Another mistake that you normally make, learners, is that whenever we speak of fovea centralis or the yellow spot, when they ask you why is the clearest image formed there, learners normally say it has high concentration of rods and cones, which is incorrect. Fovea centralis or the yellow spot has high concentration of cones. Then you have your retina, that's where you find your photoreceptors, your rods and cones. I normally say cones color. Then you have your sclera, you have your cornea, you have your conjunctiva, you have your aqueous and the vitreous humor. So remember the functions of all those parts, and especially when it comes to the eye. That's where now also you can get questions that has to do with structural suitability. They can ask you a question, grade 12 learner, to say, how is the cornea structurally suited to perform its function? Now you need to zoom in and look at the structure of of the cornea. You can actually say, because it is evenly kept, it is able to do what? To refract light. They can ask you, how is the lens structurally suited to perform its function? You need to think of what? The fact that it's transparent, it's a cause. It is able to do what? To allow light to pass through. The fact that it is elastic, it is able to change its shape in order to be able to see a far or a near object. So remember such when it comes to this, to the eye as well, the structural suitability. Having said so, I spoke about the cornea to say, they can ask you about the structural suitability of the cornea. They can even ask you about the structural suitability of the sclera. The fact that it's tough and inelastic, it is able to protect the inner parts of the ear and also to maintain the shape of what? Of the eye. Moving on, the concept, a process, pupillary mechanism. It is a reflex what? It's a reflex action. Be able to discuss pupillary mechanism is either under bright light or under dim light. As we have said before, the context will always change. The scenario will always change. You will hardly get a question, grade 12 learners, that will say, discuss pupillary mechanism under bright light. You know, they will always give you a scenario that indicates that now we are talking about a bright light, or a scenario that indicates that now we're talking about a dim room. You know, they can give you a scenario that Hotato Miss Litlotlo was recording in the studio. You know, recording in the studio, I'm surrounded by lights here, so you can tell then what needs to happen. So that simply means my circular muscles needs to contract, my radial muscles must relax, because now I'm in a bright room. The end result must be what? Less light must enter the eye. So the pupil will constrict. Grade 12 learners, remember as well, especially to learners that English is not the first language or their home language. Do not confuse the terms as well, to contract and constrict. Whenever we speak of constricting, we're talking about what? Becoming small. When the pupil becomes small. But please refrain from using the term the pupil becomes small. Remember, as I said in the beginning, science, life sciences, it's a science subject. So the correct terminology, or the use of correct terminology, is very important. So at the end, what is going to happen? You are not just going to say light enters the eye. That is incorrect. Less light must enter the eye. So that is why you can use CCRR, PCLL. What are we saying here? We are saying circular muscles will contract, radial muscles will relax, the pupil will constrict, and less light will enter the eye. Less light will enter the eye. Good. Then again, something that I wanted to bring to your attention, and actually I'll remember it later, it slipped my mind, to say, remember, oh yes, circular muscles and ciliary muscles. Do not confuse those two. That is why grade 12 learners, by tomorrow, when you are preparing today, preparation is everything, and do not confuse yourself with too many resources and too many textbooks. Just focus on what your teacher advised you to use. The DBE textbook is also a way to go. Mind the Gap, beautiful resource as well. Terminology list on our Google Drive, use that terminology list. We have provided you with previous question papers and the marking guideline. Use those previous question papers, because just starting using a textbook is not enough, and it will never be enough. So do not confuse circular muscles and ciliary muscles. That is why it is very important when making your notes, summary of your notes, just write which parts are responsible for pupillary mechanism. We're talking about circular, radial muscles, the pupil, very important. And another thing that I forgot to mention, we have other circular muscles in the part of our body. It is very important that when discussing pupillary mechanism, you need to tell us that we are talking about circular muscles of the iris, but you are not saying, as long as you have mentioned the iris at the beginning, there is no need to always mention the iris as you move on. You cannot now say circular muscles of the iris contract, radial muscles of the iris will relax, Mention the iris once, it's okay, it is sufficient. Then, if you know what happens under bright light, what happens under dim light, it is just the opposite. That now circular muscles will relax. What happens when the circular muscles will relax? Circular muscles and radial muscles, they work in opposite to one another. So if the circular muscles will relax, that simply means the radial muscles will contract. The pupil, we're not saying the pupil will become bigger. We use the terminology as well. It will dilate. And what happens when the pupil dilates? That's when now more light will enter the eye, because now you are in a dim room. Accommodation. Terminology. What is accommodation? We're talking about the ability of the lens to change its shape in order to be able to see a far or a near object. That is accommodation. Also, if you know what happens when you are looking at an object that is more than 6 meters away, that is a far object. An object that is less than 6 meters away, that is a near object. They can give you a scenario that when the person is reading a book, they don't need to tell you that when reading a book, we're talking about a near vision or an object that is near. Then what happens? That when we are talking about less than 6 meters, what happens to the ciliary muscles, and which parts of the eye are involved in accommodation? Very important. Sometimes examiners, they won't ask you for the entire process. They will be specific. They may ask you about the role of the lens when it comes to viewing an object that is near. They may ask you maybe about the role of the ciliary muscles or the role of the suspensory ligaments. So you need to remember that as well, to say sometimes it's not about memorizing read with understanding, because they may ask you only a specific part when it comes to the entire process. Lovely. So now ciliary muscles will contract, suspensory ligaments will slacken. Remember, these are ligaments, they are not muscles. They either slacken, or if they do not slacken, they do what? They are taut or they tighten up. And think about it, grade 12 learner. The minute the ciliary muscles contract and the suspensory ligaments loosen, what will happen to the tension on the lens? The tension of the lens will decrease. So when the tension on on the lens decreases, what happens to the convexity of the lens? Then that simply means the lens will become more convex. And when the lens is more convex, remember that when the lens is more convex, the refractive power of the lens increases. When the lens is less convex, the refractive power of the lens decreases. Remember that as well. So when the refractive power of the lens increases when looking at the near object, what is going to happen? The last bullet is always the same, whether you are discussing a near vision or a far one. A clear image is formed on the retina. A clear image is formed on the retina. So what is important here, grade 12 learners, is that remember the parts that are involved in the process of accommodation. Your ciliary muscles, suspensory ligaments. After suspensory ligaments, what needs to happen? You must talk about the tension of the lens. After the tension of the lens, you must talk about the shape of the lens, whether it's more convex or it's less convex. Then, depending on the convexity of the lens, of how convex the lens is, the refractive power, then you talk about the ability of the lens to refract light, the ability of the lens to bend the light. So the closer the object, the more the light must be bent. The farther the object, the less the bending of the light. Beautiful, lovely. Now let's have a look at the questions as well. So that is why I said you study the concepts, you go to the questions. You study the concepts, go to the questions, but make sure that you are fully prepared for tomorrow. Grade 12, you've been doing very well this year. You did incredibly well in term 1, in term 2. Paper 1, September paper, it wasn't an easy paper, but the overall percentage of Gauteng, it was lovely as well. So it shows that you have what it takes to make Gauteng proud. Now let's have a look at the DBE major paper 1 of 2024, looking at the concept that we've just done now. It says here, identify the part. Which part is it that you need to identify? You need to identify part A, which is the blind spot. Speaking of the blind spot, that's now we talk about that area that has no rods and cones. And we know that if it happens that the light falls on the blind spot, no image is going to be formed because there are no what? There are no photoreceptors. C, which is the cornea. And lastly, D, which is the sclera. Please remember also to look at what? At how are these structures, especially your C and D, structurally suited to perform their function. They can even ask you grade 12s about the difference between the yellow spot and the blind spot. Remember such as well. Describe how the muscles in the iris enable the person to see in dim light. How the muscles in the iris enable the person to see in dim light. Think of the end result to say, in dim light, what must happen? In dim light, more light must enter the eye because the room is dim, isn't that so? So if more light must enter the eye, what must happen to the pupil? The size of the pupil must what? Must dilate. And having said so, the radial muscles must contract, circular muscles must relax. Name the process that occurs in the eye when a person focuses on objects at different distances. We're talking about accommodation. Grade 12 learners, my humble request, study terminology. I know it's one aspect of in life sciences that you do not like, but the minute you know your terminology, you know your content, because terminology cut across. You'll find terminology in section A, you'll find terminology in section B. So the minute you're comfortable, the minute you know your terminology, then you'll be confident to say, you are ready. So before starting to study, if you're writing today, you took a break, you start with terminology. Where are you going to find this terminology list? If you haven't received one from the teacher, you have our Google Drive link. Go to the Google Drive link, you will find the terminology list. Explain how the shape, we said the process is accommodation. Explain how the shape of part B enables a person to read a book. That's what I told you about. This is the process of accommodation when looking at what? At a near object. But they do not want the entire process, Homo sapiens. They only what? They only need to understand the role of what? Of part B, which is what? Which is the lens. So you can't start telling us about the ciliary muscles and suspensory ligaments and the tension. That is not what the question is asking. Your discussion must start with from the lens to say what is going to happen. The lens is more convex. So when reading a book, because you're looking at an object that is closer, when reading a book, your lens becomes more convex. And I said to you, the more the convex the lens, the more the refractive power of the lens. So that simply means more light will be bent, and a clear image will be formed on the retina. So it is a cause and an effect as well. It's a cause and an effect question. Another beautiful concept grade 12 is the ear. Learners sometimes find the ear to be difficult because of many parts and difficult terms, because the language that we use in life sciences, it's a scientific language. It is abstract in nature. It is not the language that is used every day. That is what makes our subject a little bit challenging, but it is not just a challenging subject. It is a science that is alive. It is a beautiful subject. Now let's have a look at the ear. One thing that I need you to remember about the ear is also know the structure, know the function, know the structural suitability, know how the pinna is structurally suited to perform its function. Know how the tympanic membrane and the oval window, their role in the amplification of sound. The role of ossicles in the amplification of sound. The fact that the ossicles are arranged from the largest, which is the malleus, to the smallest, which is the stapes. The vibration gets to be concentrated, and the minute learners that when vibrations are concentrated, that simply means sound is what? Is amplified. The sound wave enters the middle ear through the large surface area of the tympanic membrane. And these vibrations leave the oval window of the middle ear to the inner ear through a small surface area of the oval window. The difference in the surface area of the tympanic membrane and the oval window, they concentrate sound. Hence, sound gets to be amplified. They can also ask you the structural suitability of what? Of the semicircular canal. How are the semicircular canal structurally suited to perform their function? Think of how the canals are positioned. They are positioned at right angle to each other. Why are they positioned at right angle to each other? So that they can detect movement coming from all directions. So please know such information. It's very important for you to ace. Actually, you are going to ace tomorrow's paper. We have so much confidence in you and our teachers as well. So know the different parts of the ear. Another important thing, learners, whenever we speak of the outer ear, we're talking about sound waves. The pinna will trap and direct sound waves through the auditory canal. The minute the sound waves hit the tympanic membrane, the tympanic membrane will start vibrating. And the vibration, so whenever we speak of the middle part of the ear, now we're talking about vibrations. You cannot be talking about sound waves. From tympanic membrane to oval window, we are talking about vibrations. When these vibrations enter the inner ear, in the inner ear, what is it that we have there? We have endolymph, the fluid called the endolymph. When this fluid called the endolymph starts moving, it creates pressure wave. So that simply means inside, in the inner ear, now we're talking about pressure wave. And these pressure waves are the stimuli. They are the ones that will stimulate the organ of coating in the cochlea. And remember, whenever a receptor is stimulated, what happens? the stimuli will be converted into a nerve impulse and be transmitted. And in this case, we're talking about where hearing is interpreted to the cerebrum. Another important thing, learners, is that whenever we speak about hearing, think of the movement of the fluid in the cornea. You must know that we're talking about hearing. But whenever we speak about the movement of the fluid in the semicircular canal, you must know now that we're talking about what? We're talking about balance. Okay, lovely. Now, let's have a look at, there are two ways in the inner ear, that regulates balance. So remember, hearing from the pinna to the cerebrum, the different parts, and the nerve you're talking about what? The auditory nerve. We're talking about the auditory nerve. Then the organ of Corti will be stimulated by the pressure wave, which is the movement of what? Of the fluid in the cornea. It's not just the movement of the fluid, it's the movement of the fluid in the cornea. Then this movement of the fluid in the cornea will stimulate the organ of Corti, which is the receptors in the cornea. Whenever a receptor is stimulated, it converts a stimuli into a nerve impulse. Then that nerve impulse will be transmitted through what? The auditory nerve to cerebrum. That's where hearing will be interpreted. But now let's talk about balance. Whenever we speak about balance, we're talking about what? Two things. We have the receptors in the ampulla, and we have the receptors in the circulus and utriculus in the vestibule. Whenever you change your direction and speed, the minute you change your speed and direction, you are stimulating what? The receptors in the ampulla, the cristae. Whenever you change, and let me just correct myself here. It's not just the change in speed and direction. It's speed and direction of the head. And change in the position of the head. Whenever the position of the head is changed with regard to gravity, now you are stimulating the maculae. Whenever the cristae and maculae are stimulated, what happens? There are receptors. Receptors convert a stimuli into a nerve impulse. This nerve impulse will also be transmitted through the auditory nerve to cerebellum. Cerebellum is the part of the brain that restores balance, will send the impulse to what? To the muscles to say, restore balance. So remember that as well, to say, look at the scenario that you are given. And from the scenario that you are given, you can tell whether what is stimulated here. Is it both the cristae and the maculae? Or is it only the cristae? Or is it only the maculae? A lovely question in our Gauteng prelim paper. A pirouette is a ballet movement where a dancer spins or rotates on one foot. The dancer's head remains still and the body turns. Very important. They are telling us that the position of the head doesn't change. When they say to us, the position of the head doesn't change, they are saying to us, the maculae are not stimulated. But the fact that the ballet dancer or the dancer is spinning, what happens is changing what? The speed and direction. So you are going to tell us that the change in speed and direction is going to cause the movement of the fluid in the semicircular canal. You can't just tell us that it causes the fluid to move. Which way? Because there is movement of fluid in the cornea that is responsible for hearing, that stimulates the organ of Corti. And also there is the movement of fluid in the semicircular canal. Then tell us that the change in speed and direction will cause the movement of the fluid in the semicircular canal. And then tell us that the cristae will be stimulated, Then the stimuli will be converted into a nerve impulse. The nerve impulse will be transmitted to the auditory nerve, to the part of the brain called cerebellum, that restores balance. That will send the message to the skeleton muscles to restore balance. So here's let's describe how the dancer maintains her balance throughout the dance. Then it says here, there is a change in speed and direction. I've spoken to this already, grade 12 learners. So please make sure that you understand this concept of hearing and the concept of balance. Now, we will just take a 2 minutes break. Then we will start with human reproduction. That is also one of the topics that has most marks and also a lovely topic in life sciences. Keep tuned on.
You're doing so well.
Welcome back, lovely learners. Now let's have a look at what? At human reproduction. As I've said, if you didn't do well, you haven't been doing well in the subject, it was just 25%. We have 75%. That is a game changer. That changes everything. And one thing that I can tell you is that you have what it takes to make it. You definitely have what it takes to make it. Now let's have a look at human reproduction. What is it that you need to know under human reproduction? The female reproductive structure, your ovaries, your fallopian tube, your uterus, your passage called the vagina, and the opening called the vulva. And also be able to tell us the structural suitability. How is the uterus structurally suited to perform its function? Very important as well. The male reproductive structure, it is very important as well, grade 12, that learn, especially when it comes to the male reproductive structure, learn both views, the front view and the side view of the structure. Having said so, do not make a mistake into going into that examination room if you don't know how to draw a sperm or an ovum. Okay, please go right. Don't say I said you mustn't go. You have to go and write. But make sure that you are able to draw the structure of an ovum. You are able to draw the structure of a sperm cell. Know the different, especially when it comes to the sperm cell, know the different functions of different parts of the sperm cell. Another structure that they can ask you under this topic to draw is the simple one, the female reproductive system. It's either they can ask you to draw the fallopian tube and the ovary or the entire female reproductive structure. So please keep on practicing. You have what it takes. You are going to do well. You've been doing very well. Then we're going to have a look at the processes Learn those processes as they are indicated in your examination guideline, your oogenesis and spermatogenesis. We call them gametogenesis. What's gametogenesis? It's basically meiosis. Puberty, the sexual maturity, menstrual cycle, very important as well. The hormonal control of the menstrual cycle, which hormones play an important role in the menstrual cycle. Then last, we are going to talk about the development of the fetus. Then my colleague Suki will take over with the other topics. Beautiful presentation is coming, I'm telling you now. Let's move forward. Male reproductive system. Know the functions of the accessory glands, your seminal vesicle, your prostate gland, your Cooper's gland. Epididymis and epidermis, those are two different things. Terminology, I'm going back to terminology to say it is very important to know your terminology. Excel in your terminology. You will thank me later, I'm telling you. Urethra, ureta. Do not make a mistake as well. There is no concession when it comes to the spelling mistake of these terms. Your auxin, your axon, your choroid, your chorion, your corpus callosum, your corpus luteum. Learn terminology. Learn terminology. Know the structure and functions of each part of the male reproductive system. I've spoken to this already. The testis, what is the function of the testis? The scrotum. Scrotum, be able to tell us why is it outside the abdominal cavity, so that it can be 2 to 3 degrees lower than the body temperature. Please, people, Homo sapiens, don't tell us that the temperature of the scrotum must be 2 to 3 degrees. That is wrong. It's lower than the body temperature. Why must that, or why must it be like that? For healthy sperm production, not just for sperm production, but for what? For healthy sperm production. Epididymis temporarily stores the sperms. Remember, do not use the term sperm duct. I told you, don't use the term sperm duct. Vast difference. That is the term that is acceptable. We are a science subject. Scientific terms. Know the structure of the female reproductive system as well. Fallopian tube. So what happens in the fallopian tube? Which process takes place in the fallopian tube? The process of fertilization. What is fertilization? The fusion of a haploid sperm cell with the haploid ovum to form a diploid zygote. Remember such as well. The vulva, the opening of the vagina. The function of the vagina, that's where the sperms are deposited, serves as a birth canal as well. Then you have your lining of the lining of the uterus. The lining of the uterus, endometrium. Please, you are making this mistake, and I say this mistake year in and year out, learners. It is endometrium, people. It is not endometrium wall. It is not endometrium lining. Stop writing that, right? Endometrium. I told you, and it's good. Now let's move on. The processes of gametogenesis, the formation of gametes by meiosis. So basically, oogenesis is meiosis in females, and gametogenesis is meiosis in males, the formation of the spermatozoa of the sperm cell. Know the process. If it means you have to memorize it for tomorrow, please do so. You need to be able to learn this process and know this process. Diploid cells in the ovary undergo mitosis. The oogenesis is the one that is a little bit longer. But remember, the diploid cells in the ovary, they undergo mitosis to form many follicles. Sometimes use like numerius. You may forget numerius. Numerius has many follicles. Then we will wait for puberty to take place. As soon as puberty kicks in, as soon as puberty starts, is the same thing saying at the onset of puberty, under the influence of what? Of follicle stimulating hormone. Let me talk about this as well. Abbreviations, acceptable abbreviations can be used anywhere in the paper except for terminology. When you ask you a question saying, The hormone that is responsible to stimulate the development of a primary follicle into a Graafian follicle. And the question is under terminology 1.2. You cannot say to us it's FSH. It's not acceptable. It's follicle stimulating hormone. You write the entire term. Then on the onset of puberty, what happens? Please do not say one follicle. It's one cell inside the follicle. That's where you're also making a mistake. You are saying one follicle. It's not one follicle. It's one cell inside the follicle that will enlarge and undergo meiosis. Out of the 4 cells that are formed, only one will form. Is not only one is forming a mature ovum or an ovum, but a haploid ovum. Terminology is very important. It forms a haploid ovum. Then spermatogenesis. Under the influence of testosterone, what happens? The diploid cells in the seminiferous tubules, they undergo meiosis to form haploid sperm cells. Now let's have a look at one of the important questions in life sciences, scientific investigation. Scientists conducted an investigation to determine the relationship between age and fertility in men. From there, that's where you get what? Your aim. And from your aim, that's where you get what? Your dependent and independent variable. So it is very important as well to have a look at this to say, you ask yourself a question, which factor influences the other? Does fertility in men depend on the age, or does the age depend on fertility? Will depend on what? On the age. So that simply means the fertility will be the dependent variable. This investigation was done from 1999 to 2017 using 1,294. Yes, not 110, it's 1,294. We don't just give you numbers in life sciences for the sake of giving you numbers. Those numbers, they're going to ask you about them. Men, from the age group of 16 to 65, men with occupations involving exposure to excessive heat were excluded during this investigation. The procedure was as follows. Semen was collected after 5 days of no sexual activity. A special microscope was used to determine sperm count and progressive motility. An electron microscope was also used to determine the sperm necrosis. That is, we're talking about immature or dead sperm per fresh semen. And remember, grade 12 learners, whenever we speak about semen, we're talking about the fluid from the seminal vesicle, Cooper's gland, and prostate gland, including the sperm. So semen is that fluid with the sperm. Identify the independent variable. As I've said, is age. Dependent variable is the fertility. State 3 ways in which the dependent variable can be determined. This is exactly taken from what you are given. The sperm count, progressive motility, and sperm necrosis. Give 2 reasons why the investigation was considered reliable. Reliability. Generally, you will say increase the sample size, repeat the investigation. That is how you generally answer a question on reliability. But here, you need to you apply the information that we have given you. The investigation was conducted from 1999 to 2007. So that simply means, from 1999 to 2017, not 2007, my apology. Then, so that simply means it was conducted over a long period of time. You cannot say the sample size was large. No, tell us that they used 1,294 men. Explain why men with occupations involving exposure to excessive heat cause an effect as well, so that the age will be the only independent variable. We do not want any other variable to influence our results. High temperatures can affect the fertility, therefore decreasing the validity of the result. Now, let's have a look at the menstrual cycle. I'm just going to summarize the menstrual cycle. Know the ovarian. Whenever we speak about menstrual cycle, we're talking about the changes that are taking place in the uterus, and the changes that are taking place in the ovary. So it is very important of you to know which hormones are ovarian hormones. We're talking about estrogen and progesterone. Which hormones are secreted by the pituitary gland is FSH and luteinizing hormone. What is the function of these hormones? Follicle stimulating hormone. It stimulates the development of a primary follicle to become a Graafian follicle. The function the estrogen, it thickens the endometrium by making it more vascular, more glandular. The function of progesterone is not to maintain pregnancy, it's to further thicken this endometrium. Then we have the luteinizing hormone that will rupture the Graafian follicle and stimulate ovulation, and also convert the empty Graafian follicle into what you call the corpus luteum. So be able to please learn your menstrual cycle to say, I'm summarizing now. From your primary follicle, for this primary follicle to develop and become a Graafian follicle, we need pituitary gland to secrete follicle stimulating hormone. This Graafian follicle cannot just release this ovum. It has to release estrogen first to go do what? To go thicken the endometrium. The minute the endometrium is thickened, then high levels of estrogen stimulate the pituitary gland to secrete LH. That luteinizing hormone is the one that is going to stimulate ovulation. Then know what happens if fertilization took place and what happens if fertilization did not take place. If fertilization took place, the corpus luteum will remain intact, its size will not change, and the progesterone will remain high. But what happens when fertilization did not take place? The corpus luteum will start degenerating. When the corpus luteum degenerates, what happens to the level of progesterone? The progesterone will decrease. And the low levels of progesterone will also stimulate the pituitary gland to secrete FSH to say, the ovum was not fertilized. Let's start developing another follicle. That is your menstrual cycle. That is your menstrual cycle. And the diagram below represents some changes that occur in the human body during the menstrual cycle. Identify A. The hormone A is estrogen, is the hormone secreted by the Graafian follicle. On which day of the cycle did the ovulation take place? Is day 14. It's not always day 14. You look at the entire cycle. Explain the negative feedback between A and C. That is follicle stimulating hormone and progesterone if fertilization did not take place. Important as well to say what happens if fertilization did not take place. It's a cause and an effect. The corpus luteum does not degenerate. So it says if fertilization took place. So that simply means it continues to secrete progesterone, or the progesterone remains high, inhibiting pituitary gland to secrete FSH. We do not want FSH to develop another follicle while the ovum is fertilized. Lastly, fertilization. I've spoken about fertilization already. Development of the zygote. From the zygote, it will develop into a ball of cells called morula. Morula will go through mitosis as well to develop into a hollow ball of cells called the blastula or blastocyst. It's not a blastocyte. It's a blastocyst. So preferably use the term blastula. Then it will develop and become an embryo. Gestation, the period of development from the embryonic stage till birth. So understand what happens as well. The function of the placenta, the function of the chorion, the amnion, the chorionic villi, and the umbilical cord. And also with the umbilical cord, understand the function of the umbilical artery and the umbilical vein. Having said so, grade 12 learners, all the best for tomorrow. We'll just take a minute break, then my colleague Suki will take over. Thank you very much. All the best for tomorrow. Do your best. You have what it takes.
Good day, grade 12 learners. Welcome to the next half of the revision for paper 1. We're going to be looking at endocrine system and homeostasis. You don't need to be scared for this section. It usually appears in your investigative questions. But believe me, learners, if you know the endocrine glands and what the hormones will do, you will be able to understand these questions. Okay? So first of all, you must know the difference between the endocrine system and the nervous system. These are two coordinating systems that regulate the processes in our body. Okay? Endocrine system deals with the hormones. And remember, the nervous system, the parasympathetic and the sympathetic, also regulate certain processes in our body of how we react to stimuli. So the endocrine system basically is the hormones that will help us to react to certain internal stimuli, and whereas the nervous system helps our body to react to external stimuli. So internal stimulus, the hormones will do the function. And external stimuli, it's the nerve that will help you. And the word homeostasis, as my colleague has indicated, terminology is vital. Homeo on its own means home. And what is the home? The internal environment in our body, which is your body cells and the fluid surrounding your cells, which is the tissue fluid, as well as your blood. Those three parts, your blood, your cells, and the fluid around the cells, they must have a constant environment of certain factors. And we need to ensure that this internal environment must be kept constant. And what helps to regulate to keep it constant are the hormones. Now, what's very important is that endocrine glands and the homeostasis part has 34 marks, which comprises of 23% of paper 1. And that's a lot of marks. If you need 30% to pass, out of the 30, 23% is endocrine glands and homeostasis. Now, let us look at some of the glands and what they do. First of all, you need to know, learners, that the endocrine gland is a gland that will secrete your hormone, okay? What is the hormone? It's a chemical messenger. It's a message that is going in your blood, but it's in the form of a chemical, a substance, and it's traveling in your blood, where directly into the blood. So endocrine glands secrete hormones, which are chemical messages that travel in the blood, right? In the blood. Don't say in the blood vessel. They travel in the blood. And then the blood carries that hormone to a specific place, the target organ. So hormones are not just released like that. They are only released when there's a need to be released, when they have to react to an internal stimulus, and they need to go to a specific place. So if the water pipe bursts in your area, you cannot go to the police station. You need to go to the municipality to report for them to fix the pipe. So if there's a crime, you go to the police station. Same way, if there's a water imbalance, then the pituitary gland needs to be notified to release a specific hormone. And that's how we are going to work with hormones. So remember, endocrine glands are ductless. There are no ducts. Whereas exocrine glands have a duct, like your sperm duct. Like, for example, your salivary gland, that's an exocrine gland. Okay, your pancreatic duct, that's an exocrine gland. But when a gland secretes hormones and goes directly into the blood, there's no ducts, it's ductless. Okay, because they travel into the blood. Now, what are some of the factors that needs to be kept constant in our internal environment? These are certain factors. For example, thyroxine, which is very important. It's produced by your thyroid gland, and you need to know the function that it regulates or it controls the metabolic rate. And know what's metabolic rate? Very vital, important, that it controls the rate at which glucose is used by your cells during cellular respiration to produce energy. So the more active you are, you're going to use more glucose. More glucose is going to be broken down, right, by cellular respiration to produce more heat and more energy. So that's thyroxine. That's how it controls the metabolic rate. And it's also responsible for the proper development of your nervous system. Right, what other factor must be kept constant? Glucose. Glucose is what gives you energy. You don't have petrol, you're not being charged, you don't have batteries. So what's your fuel to give you energy? It is glucose. And where do you get glucose from? All your carbohydrates, your bread, your rice, your sugars, your cold drink, your sweets, your candies. That gives you glucose. And that glucose cannot be high. So no matter how much of sweet things you eat, the glucose in your blood must be at a certain norm. And when it goes above normal or below normal, then that imbalance has to be is detected now by these glands, which are going to secrete hormones to regulate that glucose, which is below, to come back to normal, high to come back to normal. Okay? So glucose must be kept constant. It mustn't be too high or too low. Then salt. We need salt in our blood. Salt keeps water. Salt retains water. But you can't have too much of salt, because if salt retains water, too much of salt is going to retain too much of water, and it's going to cause your blood pressure to increase. But on the other hand, you can't say, I don't want to eat salt because my pressure is going to increase. Then you don't end up eating salt, and then you don't have enough salt to retain water, and the pressure is going to go very low, and then you're going to start fainting. So the salt has to be within a normal limit. If it's too much, then we have a hormone to control it. If it's too little, then the hormone is going to do something else to make sure it comes back to normal. So salt. Water. We need water. You cry. How are you going to cry if you don't have water? You need tears, water, saliva. It needs water. Your blood needs water. How is it going to flow? So water must not be too much. You can't say, I need water, so I must over drink the water. No, it must be a certain limit. Remember, we have animal cells. We don't have a cell wall. So if you have too much water, they can actually burst as well. So the water is regulated, by ADH, right? And the hypothalamus. So water must be kept constant. During COVID, they should take your temperature. Our body core temperature must be 37.5 degrees, irrespective of whether it's -2 degrees outside or whether it's 25 or 35 degrees outside, whether it's boiling hot or freezing cold. Our temperature, our body core temperature must be 37.5 degrees Celsius. If not, it's going to affect all the enzymes in the mitochondria that control respiration. So if your temperature goes too high, the enzymes will denature. The enzymes what? That control digestion, the enzymes that control respiration, they'll denature. Your glucose won't be broken down. You won't have any energy. You'll be cold. You'll be dead. Okay? So therefore, your temperature has to be constant, 37.5. You can't also freeze. It can't also drop. Because if it drops, the enzymes become inactive temporarily. And then they won't be able to control respiration, and no heat is going to be produced. Carbon dioxide is what we exhale as an invisible gas. And that carbon dioxide is coming from where? It's coming from the breakdown of glucose during cellular respiration. So the more active we are, the more glucose is going to be broken, because we're active. We need more energy, more glucose. More glucose will be broken down by cellular respiration to produce more heat. So your temperature is also going to increase because you are active, you are busy running. Your temperature is going to increase. You're breaking down more glucose. You're producing more energy for your muscles. But at the same time, glucose is made up of carbons. That carbon is going to be released. And you can't have too much in the blood. It's going to react with water in your blood to form carbonic acid and decrease your pH. And that can cause your enzymes also to denature because they're sensitive to pH. So we have to keep the carbon dioxide in the blood very, very low. And we're going to see the mechanism to do it. Right, now learners, this is what we call a flow diagram. And be very careful. If they ask you to draw a flow diagram, you don't go and draw the pituitary glands and all that. You use words and arrows to draw a flow diagram. If they ask you to describe, you write in a paragraph, and don't separate your paragraph. You put all the facts in one paragraph so that the person who's reading your answer can read and give you allocate the marks. So don't start dividing your paragraphs. So only if there's a flow diagram, you will write words and arrows to do a flow diagram. This is how the thyroid gland controls thyroxine. So let us see. If your thyroxine level is below the normal limit, so when something is below the normal limit, we need something to happen to increase it to the normal limit. And that is called a negative feedback mechanism. So learners, just remember, to keep things constant, we need what we call a negative feedback mechanism. What is a negative feedback mechanism? It's when certain factors are above or below the normal limit, then another factor will have the opposite effect to bring it back to normal. When certain factors are above or below the normal limit, then an opposing factor will bring about, or another factor will bring about the opposite effect to bring it back to normal. That's called a negative feedback mechanism. So when your thyroxine level is too low, right, then the low level of thyroxine will be an internal stimulus. It will send a nerve impulse where to the pituitary gland that lies below the hypothalamus and stimulate the pituitary gland to secrete what? More TSH, because your thyroxine is low. So the pituitary gland will secrete more thyroid stimulating hormone so that the thyroid gland can be stimulated even more to secrete more thyroxine because the thyroxine is low. So here the pituitary will secrete so low levels of thyroxine, pituitary secretes more TSH, and then the more TSH or the high levels of TSH will stimulate the thyroid gland to produce more thyroxine, and the thyroxine level goes back to normal. Now, how will you apply this knowledge to a question? They would say a person went to the hospital to do a blood test, and they found in the blood test that all the time his TSH levels are very high, higher than normal. What does it mean? It means that if the TSH level is higher than normal, it's like the pituitary gland is trying to constantly activate the thyroid to secrete more thyroxine. It means that his thyroxine level is very low. That's why the TSH is remaining high, because his thyroid gland needs to produce more thyroxine. His thyroid gland is probably underactive. Okay, and remember, the disease that's caused when you have low levels of thyroxine is when your thyroid gland swells up. It's called goiter. All right. So what happens now when your carbon dioxide level goes low or high? Remember now, learners, what produces too much of carbon dioxide is when you are very active. Now, here is the flow diagram, right? When the carbon dioxide level in the blood increases, right, it will decrease the pH level. Right, so when your carbon dioxide in the blood rises, it's going to stimulate a receptor where? In your carotid artery in the neck. And this receptor is going to send a nerve impulse to the medulla oblongata, right, which is just above the spinal cord. And the medulla oblongata now is the center that's going to send a nerve impulse to your breathing muscle, which is your diaphragm and your intercostal muscles. And they will contract and relax actively, more active. Right? Increase inhalation, increased exhalation. So your breathing will be faster. The medulla oblongata will send a nerve impulse to the heart. Right? The heart will beat faster so that more carbon dioxide, more blood with more carbon dioxide will be sent to the lungs to be exhaled. And then the CO2 level will go down. So when the carbon dioxide level in the blood increases, right, the receptor cells in the carotid artery will be stimulated. Impulse will be sent to the medulla oblongata. The medulla oblongata sends the nerve impulse to your breathing muscles, which is your diaphragm and your intercostal muscles. The breathing becomes more active, inhalation and exhalation will increase, and then the heart will beat faster because the medulla oblongata sends the nerve impulse to the heart. And what does it cause? It's going to cause the heart to beat faster. And what is the effect? Is that more blood will be sent to the lung. So more CO2 will be exhaled. More blood will be sent to the heart, right? Also for the heart to pump more blood with CO2 to the lungs. And there you see more carbon dioxide is taken to the lungs and will be exhaled faster, and the CO2 level goes back to normal. Now you need to know the term osmoregulation, which means that's the term we use to control the water levels in the blood. Now, what happens if you are not drinking sufficient water, or if you are sweating on a hot day, you are losing water. So that means that the water level in your blood, in your tissue fluid is dropping. So you cannot afford to lose more water in your urine as well, because you're already losing water through sweating. So that low water level in the blood is an internal stimulus. It will send a nerve impulse to a specific center, which is your target organ. And that target organ is going to be the hypothalamus. The hypothalamus now has receptors called osmoreceptors. They are able to pick up that the water level in the blood is now low, because the person is sweating. So the hypothalamus will now, the receptor receive that water level is low, then the osmoreceptors in the hypothalamus stimulate the pituitary gland to secrete ADH, capital ADH, which is antidiuretic hormone. The ADH now is a hormone, and now it's going to go to a target organ. It's going to go directly to the blood because it does carry the message fast. Go directly to the target organ called the kidney. The kidney is the organ. But where in the kidney? It goes to the kidney tubules, which are called renal tubules. And what does ADH do? Now, normally the renal tubules don't allow water to pass through them. It keeps the water there to obviously form urine. But when your water level is low in the blood, you can't afford to pass more water in your urine. So the ADH actually is going to make the kidney tubules permeable to water. It's going to allow water to move out of the kidney tubules and to be reabsorbed back into the blood. Do not say absorbed, because water is already absorbed in your large intestine. Now it went back into the blood, and now it filtered into your kidney. Now that water needs to go back into the blood because your water level in the blood is low. You're sweating. So what does ADH do? ADH makes the kidney tubules permeable to water so that more, not just water, so that more water is reabsorbed into the blood and the water levels go back to normal. This is called osmoregulation. And how will it have? What effect will it have on your urine? The urine is going to be concentrated, concentrated, because there's less water in there, okay? And obviously, if you drink lots of water, the water level is high, then it will stimulate the pituitary or the hypothalamus to secrete less or no ADH. The kidney tubules will remain impermeable and more water will be excreted in the urine because you have a high water balance in your blood, okay? Now we're looking at glucose. Learners, there cannot never be a paper one without glucose. Okay? Insulin and glucagon. Pancreas is the organ. You know what happens here. This is a little flow diagram to summarize, to understand the workings of insulin and glucagon on glucose. So here's the normal glucose level. And what happens here is they say, if the glucose level increases what? It increases above normal. You mustn't just say it increases. If the glucose level increases above the normal limit, then that is an internal stimulus. It's going to send what? A nerve impulse to the pancreas. And the pancreas is now going to send what? The pancreas is going to secrete by the islets of Langerhans, so the beta cells, is going to secrete insulin, which is the hormone. The insulin now will now go directly by the blood, remember it's a hormone, to the liver. The liver is the target organ. And the insulin is going to stimulate the liver to convert the excess glucose to glycogen. Not just glucose, but the extra glucose, because the glucose level went above normal. So it's going to stimulate the pancreas to secrete insulin, and the insulin is going to convert the excess glucose to glycogen in your liver. Okay, so insulin stimulates the liver to convert excess glucose to glycogen, and then the glucose level comes back to normal. But now let's say you've been fasting and you haven't been eating, so your glucose level goes lower than normal, decreases lower than normal. Then the low levels of glucose is the stimulus, which will send a nerve impulse to the pancreas to secrete what we call glucagon. Don't get confused with the words glucagon and glycogen, right? So glucagon, learn that spelling. It's a hormone, and it's going to, what is it going to do? Glucagon is now going to convert or stimulate the liver to tell the liver, you had stored glycogen. Now convert the stored glycogen back into glucose, and the glucose level will rise back to normal. Right, now we're going to look at salt. Salt hormone is aldosterone. Right, and what is the organ that's going to control the salt? Also the kidney tubules. So let's look at this. Here's the normal salt level. If your salt level in the blood decreases, now you're not eating much salt, then what's going to happen is that the low salt level in the blood will send a nerve impulse to the adrenal gland. It's the gland above, on top of the kidney, not above the kidney, on top of the kidney. Above means it's away from the kidney, but on top of the kidney means the adrenal gland is on top of the kidney. So a nerve impulse is going to be sent to the adrenal gland, and the adrenal gland is going to secrete a hormone called aldosterone, right? And now, because your salt level is low, so aldosterone is going to go straight to the kidney tubules, and it's going to make the kidney tubules permeable to salt, so more salt will be reabsorbed into the blood because the salt level is low. So low salt level, more salt will be reabsorbed into the blood from the kidney tubule. But who's going to do it? It's aldosterone, because salt just can't move on its own. Aldosterone is a chemical messenger instructing the kidney tubules, please allow salt to be reabsorbed into the blood. Let it become permeable. So salt leaves the kidney tubules to be reabsorbed into the blood, and the salt level increases to normal. If your salt level is below normal, that means your salt level is higher than normal. Salt level is higher than normal. Probably you have too much of salt into your popcorn. So what's going to happen there? The high levels of salt will be a stimulus which will stimulate the adrenal gland to secrete less aldosterone because you got enough salt. You don't need to reabsorb more. So the adrenal gland will be stimulated to secrete less or no aldosterone. No salt will be reabsorbed from the kidney tubules into the blood, and more salt will be excreted in the urine, and the salt level goes back to normal. Homeostasis is maintained regarding salt. Thermoregulation is a very important word, learners. It means controlling the temperature. So if there's an investigation on the environmental temperature, on the effect of body core temperature, remember now, you need to keep a temperature constant. So controlling the temperature is called thermoregulation. And what controls it is one organ called the skin. Which two parts of the skin controls it? It's the blood vessels and the sweat glands. That is what's in our syllabus. Right. You can see on a hot day, the sweat gland becomes more active. And when it becomes more active, we're going to produce more sweat. And what's going to happen? By sweating, the sweat, when it evaporates, it cools the body and brings the temperature back to normal. Another thing what happens is with regards to the blood vessels, on a hot day, more blood flows to the surface of the skin. So more heat is lost by convection. So you are controlling your body temperature in two ways, where more blood flows to the surface and then more heat is lost, right, by conduction. And then also the sweat glands become more and you secrete more sweat. The sweat evaporates to cool the skin, and that's how your temperature comes back to normal. On a cold day, and also remember, learners, when the more blood flows to the blood vessel, it's called vasodilation. Now, on a cold day, you're shivering. You can't allow more blood to flow to the surface. You start losing heat. It's already so cold. So what's going to happen on a cold day? The stimulus, it's cold. Then it's going to send a nerve impulse where? To the hypothalamus in the brain. The hypothalamus now will send a nerve impulse to your sweat glands and blood vessels. And on a cold day, it will tell the sweat glands become inactive, no sweat is secreted, no evaporation. Blood vessels become constricted, less blood flows to the skin, and more heat is retained in the body. Okay, do not say that the blood vessels move to the surface of the skin. It's that more blood moves to the surface of the skin, okay? Or less blood flows to the surface of the skin. That's vasoconstriction. More blood flows to the surface of the skin because the blood vessels are dilated. That's vasodilation. So hot days, vasodilation. Cold days is vasoconstriction. Cold and constriction with a C. Okay, now let's look at some questions here, learners. You did see this question in your paper. Very good question. All you need to know is that if a person is diabetic 1 or diabetic 2, it means that the body is either having no insulin, as in diabetic 1, or diabetic 2, there is insulin, but the body cells cannot react to the insulin. So basically what's going to happen is that we know insulin controls the blood glucose level. So whether you're diabetic 1 or diabetic 2, your glucose is going to be out of control. Okay, so now, if the person is a diabetic patient, that means that the glucose level is above outside the normal limit. In response to the alert, you remember now you can either prick your finger to test your glucose, or you can put a sensor, which they called the continuous glucose monitor. They stick it like a little plaster on the skin, and it detects the glucose level in your tissue fluid, right? Now, if it beeps on your cell phone, it's connected wirelessly to your cell phone. If it beeps, what does it mean? The glucose level is outside the normal limit. In response to the alert, she immediately injects herself with the required dose of insulin. State whether the smartphone alert warns her that the blood glucose is going to be high or low. Obviously, it's going to warn her when it's high, okay? Because it's high, monitor the glucose if it's too high. Can you see that? Right. The next question, same paragraph. But a different question. Describe the role of the injected insulin in regulating her blood glucose level. When it is explained or described, you must have a cause and effect. Understand what is the purpose of her injecting insulin, right? And what's going to do? Insulin will stimulate the cells to absorb more glucose for more respiration. Okay? And also what the insulin going to do? It's going to convert the excess glucose into glycogen and store it in the liver, and by that way, by using more glucose in the cells, where the cells absorb more glucose for respiration, by converting the excess glucose to glycogen, it will bring her blood glucose levels to normal and regulate it. All right, now this got to do with adrenaline. You can see the boy is running. This adrenaline hormone got to do with flight, emergency reactions. It works the same way as the sympathetic nerves, which belongs to the autonomic nervous system. But in the endocrine system, we talk about adrenaline, which prepares you for or stimulates you in an emergency. Right, the adrenaline, but in the nervous system is the sympathetic nerve. So name the gland that's responsible for this reaction. You can see the boy is very scared, he's running away from the dog. So that gland is going to be the adrenal gland. So the adrenal gland secretes two hormones. It secretes adrenaline for an emergency and aldosterone for maintaining the salt balance. Where is the gland located? The adrenal gland is located on top of the kidney, right? That's why it's called adrenal. All right, remember renal is kidney. Explain the effect of adrenaline on the heart. Now here, explain means cause and effect. You don't not just say it'll cause the heart to beat fast. Why? It's going to cause the heart to beat faster because it needs to pump more glucose and more oxygen to the muscles so the boy can have more energy to run away from the dog. Can you see that? That is the effect. So the respiratory system, if you're running away from the dog, are you going to breathe like normal? No. You're going to breathe very, very fast. So the rate and depth of breathing is going to increase. Why? You need to take in more oxygen for cellular respiration to produce more energy for the muscles to move. And that's going to be the effect. So you can see it's going to stimulate the breathing muscles to increase the rate and depth of breathing so that more oxygen is inhaled. For what? For cellular respiration, to produce more energy because he's running. His muscles need energy. And therefore, it's going to also, on the heart is going to beat faster to pump more glucose and more oxygen to the muscle cells for respiration. Can you see that? So it's transported even faster. And then we're going to look at plant response. Learners, plant response is not a difficult question. It's very simple, right? It's 13 marks. And then you can see the plant hormones are auxins, gibberellins, and abscisic acid. Auxins bring about tropism. Tropism means the bending of part of a plant towards the stimulus. Okay, and it's always produced at the tip of the root and the tip of the stem. Now, learners, with auxins, just remember, when you have a stem, whether the stem is in space or whether the stem is in the ground on earth, right? Or sorry, the root. Just remember, whether the root is in space or whether the root is in the ground, as long as it's a root. And you talk about auxins. Auxins, wherever they are, whether in space, there's no gravity, or whether in the ground, the auxins where there's a high concentration of auxins in a root, on that side, there will be no cell elongation and no cell enlargement or cell division. But there will be cell elongation and cell division faster on the opposite side where there's less auxins. So in a root, where there's more auxins in a root, there'll be no cell division and no cell division and cell enlargement and cell elongation. But on the opposite side, where there's less auxins, there'll be more cell division, more cell elongation, and that part will grow faster. Now let's look at the stem. Whether the stem is in space or whether the stem is above the ground on earth. If it's a stem, auxins, when there's a high concentration of auxins in a stem, then that side will have more cell division, more cell elongation, and that part will grow faster and longer and bend. Now, remember, there are two stimulus, light. If there is light coming from all sides, auxins will move, they shy away. They'll move in the center, and then they're going to cause cell elongation upwards. So as long as auxins are present at the tip of a stem, they will cause the plant to grow upwards. It's called apical dominance. So when auxins are concentrated on the tip of the stem, they are dominating the tip. They will cause the plant to grow taller. like a Christmas tree. But when we are going to cut off the tips, we're going to remove the tips. We are pruning the trees. We're removing the auxins. Then they're not going to promote apical dominance. They're going to cause more lateral branches, more side branches. The plant will become more bushy, and more fruits and more branches will form, and more fruits will be produced, increasing the yield and increasing the economy. That's how auxins will do that, okay? So just remember, auxins are causing movement towards a stimulus, right? And when they go towards light, when the stem moves towards light, it's phototropism. And when the root grows down towards gravity because of auxins, it's called geotropism. But now, when a stem falls on the ground, right, there's no light. Okay, we're not talking about light now. They just said the stem falls on the ground. Now, what's going to happen is that the auxins will move down. Now, that's on Earth. The auxins will move down towards gravity, okay? They're going to form on the lower end of the root. So where the lower end of the root, that means that part won't have cell elongation, cell division, but the opposite side will grow faster and faster and faster, and that's where the root will bend down towards gravity. But in your prelim paper now, let's look at it. Right, here is the prelim paper. They are telling you that there was an investigation where they took a germinating bead seed. Remember the stem, that's the plumule, and that was the developing root, the radical. And what they did was, they know that those two parts, the stem and the root, produce auxins. So they actually cut off that part which produces auxins, and then they injected their own auxins in the stem on that side, and they injected auxins on the root from that side. But this seedling was in space where there was no gravity. So if the auxins moved, it was not because of gravity, because there's no gravity here. So let's look at the root. In a normal condition on Earth, these auxins will go on the lower end, but they remained on the top because of no gravity. So high concentration of auxins, right, in the root here, that's where it's high, because it couldn't move down, so it remained high there. That means on that side of the root, on the top end, there's going to be less cell elongation and less cell division, and the lower end will grow faster and faster and turn up. Now let's look at the stem. There's no gravity here, right? So that's where they injected the auxins, and the auxins are on that lower end of the stem. And what we said, whether the high concentration of auxins, right, in the stem, that part will grow, have more cell division, more cell elongation, and that part will grow longer and longer compared to the opposite side where there's less auxins, right? Or no auxins. So that part will grow up and up, and then you're going to have a diagram where both the root and the stem are moving up. So what's the tropism? It's the growth movement or the bending of part of the plant towards an external stimulus. So growth of part of the plant towards an external stimulus, right? And you can see, explain the significance of cutting off the tips of the stems during this investigation. Why did they cut off the stem tips and roots? They wanted to remove the auxins because whatever movement was caused by the young root and the young stem, it must be because of the auxins they injected and not by the auxins that were already present in the stems. So they removed it because now they want to do their own investigation of how auxins will react in space. That's why they removed the original auxins, so that whatever movement there was in the root and stem, it was due to the auxins injected. See, auxins are produced in the roots of the stems and tips, and therefore if they're removed, they won't be able to diffuse, and they cannot interfere with the injected auxins. And therefore, they ensure that the effect of the auxins or the direction of the growth is only due to the injected auxins and not on the auxins that were already present. And remember now, they said, redraw this diagram of how will this root and stem be. And you can see now, because there's going to be in the root more cell elongation at the bottom, right? Because in roots, high concentration of auxins in a root inhibit cell division and cell elongation, and promote cell division and cell elongation on the lower side, on the side where there's less. So it's going to increase that way. You see it moved up. And here in a stem, high concentration of auxins in a stem will promote cell division and cell elongation. That part will grow faster and faster, and that's how you had that diagram looking like that. Okay? And then learners, reproduction in vertebrates, these are the 5 reproductive strategies. Internal and external fertilization is a strategy. Ovipary, vivipary, ovovivipary is a strategy. Amniotic egg with the amnion, chorion, and allantois is a strategy. Precaution and nutritional development is a reproductive strategy. And parental care is a reproductive strategy that contains for 8 marks. Learn the differences between ovipary, ovovivy, and vivipary. Remember that fertilization can be internal or external in any organism, okay? But mostly in fish and frogs, you find external fertilization. That means where are the gametes going to meet? Inside the mother's body, then it's internal. Outside the mother's body, if the gametes meet, like in water with fish and frogs, then it's external. And remember now, in ovipary, what does it mean? Ovipary means the eggs are laid. And do not say that the baby or the young is developing in the egg. It's not the baby or the young, it is the embryo that is developing within the egg outside the mother's body. That is, the eggs are laid and the embryo is developing inside the egg outside the mother's body. That is ovipary, where the embryo is getting its nutrition from the yolk. Right. Now, ovovivipary, it's when the developing embryo develops in the egg within the mother's body. And then it hatches or it's born alive. Then it hatches and it's born alive, okay? So it's inside the mother's body, the yolk is present, and you find that the shell may be leathery because the young is developing the embryo is developing within the egg, within the mother's body, okay? And then they hatch in the mother's body and then they are born. That's why it's vivipary, because they hatch in the mother's body, they're developing in the egg in the mother's body, that's ovo. And once they hatch in the mother's body, then they are born alive. That's like a reptile, like a snake or a crocodile or a lizard. Right. Now, vivipary is when the developing embryo develops in the uterus and gets its nutrition from a place called the placenta, like in humans, in dogs, in cats. And then the mother gives birth to the young. So the nutrition comes from the blood in the placenta. Okay. So here you can see you've had this question in your prelim paper. It's got to do with the echidna. Okay, echidna is a mammal and it uses internal fertilization. That means the male deposits sperm into the female's body, right? And then when the baby, you can see here, the newly laid egg goes into a deep pocket or pouch on her stomach to keep it safe, right? So there's a single egg that is laid. So she internal fertilization, she lays the egg. Then the egg goes in her pouch. And in her pouch, there are little mammary glands with secret milk. But that little echidna, baby echidna, has no, just skin with no spines. So as long as it's unable to move and there's only skin, no feathers or no skin or no spines, it means it's altricial development. If its eyes are closed and it cannot move, it's altricial development. Plus, remember, she had for 53 days it remained in her body, and then it only becomes independent after 7 months, which means that this echidna is an example of altricial development. All right. So tabulate 2 differences between vivipary and ovovivipary, which is the reproductive strategy, right? Internal fertilization, external fertilization, vivipary, ovovivipary, parental care are reproductive strategies. So what are the differences between vivipary and ovovivipary? First of all, to tabulate, you must draw a table and you get a mark for the table. Know what are you looking for? Ovipary and vivipary. So you put the 2 headings and then you always compare the same characteristic. So the embryo develops in vivipary. Where does the embryo develop? It develops inside the uterus. In ovipary, the embryo, can you see the embryo develops inside the amniotic egg, okay? But outside the female's body. Then, what about the food supply? In vivipary, the embryo receives a continuous supply of food from the placenta. Whereas in ovipary, you see the mother can eat right through the 9 months, but in ovipary, the yolk is fixed, just a set limited amount of food. So in ovipary, the embryo receives a fixed amount of food from the yolk. It's just there to last for a little while. So the fixed amount of food. In vivipary, the mother gives birth to the young. In ovipary, the young hatch out of the egg, right? In vivipary, regarding the waste from the developing embryo is excreted via the placenta. Whereas ovipary, waste is excreted in the allantoid. Vivipary, how does gaseous exchange occur? To the placenta, right? Ovipary, how does gaseous exchange occur? Via the amnion and chorion. Okay, vivipary, how is it protected? The developing embryo is protected by the mother's body within the uterus. But in ovipary, the developing embryo is protected by the shall. And that brings us to the end of the lesson, learners. You can do it for paper 1. Remember, there are lots of topics. Burn the midnight oil today, and you are not blank. Go through your work, be relaxed, go through it in your mind, talk about it up and down, and you'll do well. All the best. Thank you.